Hi, this is Lab 6 about glucose metabolism in yeast cells. I am Aram Nadim Asmar Cruz, an undergraduate at the University of Alberta. The title of this presentation is How the amount of glucose impacts the amount of carbon dioxide produced by the fermentation in yeast cells. First, I am going to introduce glucose metabolism in eukaryotes. The essential part of all living things is ATP. ATP, adenosine triphosphate, can be formed by the catabolism of proteins, triglycerides, and carbohydrates that will break down into amino acids, glycerol, fatty acids, and glucose respectively. And each of these can follow two processes: fermentation to regenerate NAD, and cellular respiration through the citric acid cycle to produce ATP through substrate-level phosphorylation and oxidative phosphorylation. But this last process can just be done when the pyruvate metabolism is in the presence of oxygen. When the metabolism lacks oxygen, fermentation is used, where glucose is transformed into two molecules of pyruvate via glycolysis, then into acetaldehyde and ethanol. This cycle has as its primary purpose to regenerate NAD. The main focus of this experiment was fermentation, since yeast cells get ATP through this process. Anaerobic respiration allows yeast cells to produce NADH, ATP, and waste products like carbon dioxide and ethanol. In this experiment, the amount of carbon dioxide in milliliters produced by fermentation when yeast cells are in different substrates was measured. Global warming due to the use of fossil fuels is a concern for the international community, since the extraction and combustion of these non-renewable sources pollute the environment to a great extent. In this sense, the importance of this experiment has to do with the applications in the real world that fermentation can provide us, since fermentation produces ethanol that can be used as a biofuel, an alternative to fossil fuels that are carbon neutral due to the photosynthesis of the plants before their harvest for biofuel purposes, and because they are a source of renewable energy. This study aims to determine which substrate is the best to produce ethanol through yeast fermentation, and determine how the amount of glucose in each substrate affects this process. The hypothesis is that if the substrate contains low disponibility of glucose, then we expect to observe a slower process of fermentation and little carbon dioxide and ethanol output, because the amount of glucose determines the fermentation rate. The materials used were 5 test tubes, 1 Durham tube, 6 milliliters of yeast cells, 5 milliliters of corn syrup, 5 milliliters of cob extract, 5 milliliters of kernel extract, 5 milliliters of water, 1 milliliter of amylase, water bath, vortex, Pasteur pipette, and more pipettes. For the experiment, we had 3 treatment groups and a control group. Treatment 1 involved using 5 milliliters of corn syrup and 2 milliliters of yeast cells. Treatment 2 involved 5 milliliters of cob extract and 2 milliliters of yeast cells. The treatment 3 involved 5 milliliters of kernel extract, 1 milliliter of amylase, and 2 milliliters of yeast cells. Lastly, the control group was just 5 milliliters of water and 2 milliliters of yeast cells. The procedure was adding the substrates to the tubes and vortexing them well, then warming the test tubes in a 30 degrees water bath for 10 minutes. After 10 minutes, add 2 milliliters of yeast cells to the test tubes, vortex them, and with this done, fill the Durham tubes and invert them in the containing tubes. Finally, record the volume of CO2 every 5 minutes for 45 minutes. This is the figure 1 of the production over time after the experiment. It can be seen how the control group did not react to the experiment and is a steady flat line. The experimental group 1 had the lag phase in the first 5 minutes, and then it had the log phase until the 40-minute mark, where the CO2 displaced the entire liquid volume of water. The experimental group 2 had a lag log phase in the first 5 minutes, and then it got stuck in the stationary phase for the rest of the experiment. The experimental group 3 had the lag phase in the first 10 minutes. From the 10-minute mark to the 35-minute mark, it got into the log phase, and finally the stationary phase started from the 30-minute mark. This is a figure 2 showing the production rate of CO2 of the experimental group 1, 5 milliliters of corn syrup and 2 milliliters of yeast cells. The trend line above shows the increasing pattern, and the linear equation shows the average increasing rate per minute being on average 0.104 milliliters of CO2 per minute until the 40-minute mark, since that was when the Durham tube was full of CO2. Expected, since experimental group 1, being the corn syrup extract almost entirely made of glucose, had a fast rate of fermentation in comparison with the other control groups. The scientist Flores and her team in 2018 stated that as high as the glucose concentration, so will the production of ethanol and carbon dioxide. So this explains why this experimental group had the highest rate of fermentation. The kernels are made about 70% starch, so the amylase helped hydrolyze starch into sugars in the experimental group 3. However, the proportion of amylase was low against the amount of kernel extract, and that is why there was a medium rate of fermentation. Cob extract is made almost entirely of cellulose. Even if it might contain glucose, it is a non-significant proportion. That is why the second experimental group had a deficient fermentation activity. The conclusion of this study is that the high amount of glucose in a substrate is directly related to a high rate of fermentation, therefore more CO2 and ethanol, hence biofuel. However, in means of added value, it is not enough, since the parts that contain more glucose are the same parts that humans use for consumption. More research in value-added crops should be done in order to be sustainable and have an actual innovative proposal for biofuels. Finally, I would like to thank my partners Era Sanchez and Kevnit Sahota. Thank you.